Today we are going to talk about a really important and relatively new technology that you've likely heard about, and this is the CRISPR-Cas9 system. There are also now some slight variations on this that you'll hear about, but we'll stick with the original. They all work relatively similar, so this will give you a good background to start with. Let's first start by reviewing or introducing two definitions. A protease is a protein which cuts up other proteins, and a nucleus is a protein which cuts up DNA. Both of these can be relatively general, something you'd want in, say, the case of an apoptosis cleanup crew, or it can be incredibly targeted to cut in a very, very specific sequence. And then one more reminder before we go on. Remember, it's proteins which do most of the work of our cells in our bodies, and these are made up of a sequence of amino acids. Our DNA sequence codes for this sequence via messenger RNA. Genetic diseases arise because one of the base pairs change in a way which changes the amino acid of the protein. Sometimes this doesn't change the function. Sometimes it reduces it, and sometimes it knocks it out altogether. Depending on the severity and the purpose of the protein, this can result in anything from no significant difference to small changes, to genetic diseases, or even to a non-viable person in the cases of many spontaneous miscarriages. Much of our treatment for genetic disorders before gene therapy involved finding ways around this protein disruption, or just symptom mitigation in many cases. This is problematic as it makes the condition chronic and in many cases still very harmful. In many cases, even with our medicine, the person can still die very young and have a very painful short life. Certainly not ideal. Rather than having to give medicine, a better way to fix this would be to fix the genes involved. That way, the body's own machinery would then take over and make the healthy protein, and all would be well. So it would be really convenient if we could just fix the gene, which is, of course, what gene therapy does. The problem is that most of our gene therapy techniques were a bit clunky. And of course, you can't just mutate one cell and expect it to cure the patient. What percent you need to mutate changes a bit on the disease and a myriad of other variables, but it's certainly not simple. CRISPR gives us a less clunky way of doing this engineering. So I know I had you watch some videos on exactly how CRISPR works, and I'd refer you back to those now. A non-animated lecture is simply not the best way to present this information, but I think it's worth summarizing the important points that we'd expect you to know. So I'm going to leave this slide here to do that. Of course, you can access the text in the Google folder. If you didn't watch the videos on this, please make sure you do that now. I also included another one on this slide in case you'd like another video to add to your already watched videos. Now, let's talk super quick about where this comes from. Bacteria are constantly being attacked, not just by other bacteria like we discussed back in our antibiotics lecture, but also by viruses. One aspect of their immune system allows them to incorporate viral DNA into their genome. They can then use this to recognize viral DNA and cut it before disabling it. CRISPR refers to the repeated DNA sequences. Alright, so all of that is great, but what can we actually do with it? Well, lots of stuff. It can disable genes, meaning to just ensure none of it gets made. It can edit genes to, say, fix a mutation which is causing a problem. It can increase or decrease transcription, so making more or less of it. Something that may not be quite as obvious is that you can also label genes in chromosomes, which becomes a rather interesting molecular biology technique for learning about what is happening in cells. So with all this talk about CRISPR being cheap and easy, let's check out just how cheap and easy it is. This is a video of a person walking through a do-it-yourself kit that you can do at home. Hers didn't actually go super well, but that's largely just how careful you follow the directions. If you're detail-oriented, you'd have no problem. That being said, it's a kit meant to replicate something that has already been done, just like when you walk into most lab classes in college. The hard part has really already been done for you in the development. So take a moment and watch this, and then come back when you're ready. As always, all of these links are in the slides, so you can get to them by clicking through that easily. Here's a short video talking about a rather infamous biohacker, Josiah Ziner. If you are interested in learning more on this type of thing, Netflix actually has a great documentary called Unnatural Selection. I really highly recommend watching it. I honestly thought about just assigning that instead of my video, but I didn't know how to guarantee access. So here we are instead. It talks a lot about Josiah Ziner, as well as some biohackers that are trying to do HIV treatments with it. one who is engineering his dogs, and then more legitimate scientists that are actually working in the field. It's very entertaining and very watchable. If there's an interest in it, perhaps we can schedule a Netflix watch party on it and have a Zoom call chat going on. If you're interested and I forget that I made this offer in the video, let me know and we'll set it up. But for now, we're stuck with Josiah. After watching the video, talk with your group about the video and for the specific question on this that I put here. So what I'd like you to do is just talk about why what he did was just a dumb stunt. I'm sort of implying in the question that it's not actually going to do much of anything. So that's kind of where I want your conversation to go there. So this topic has been really interesting to teach on. The first year we got to introduce it as this cool new thing that had lots of possibilities. The second, we got to introduce it as this cool new thing with all these neat possibilities that are a little bit clearer and a little more likely. And then the third, we got to look at all the phase 1 trials. And now we're actually pretty far into higher level trials. Rather than try and talk about all of these here, I'm going to instead send you off to do some exploring with your groups on what some of the companies are working on. So I picked a few companies. Go out and look at these or any other company or university that you want to that are working on different CRISPR treatments. And then pick your favorite 4 and write up a quick 1 to 3 sentence summary. What you could do, if you wanted to do this in your groups, is assign everyone in the group 1 and then report back to each other as a way of divvying up the work, but still getting to see lots of interesting applications. Perhaps many of you remember a big controversy when a scientist in China edited the genome of twin girl fetuses who were then born in China. He deleted a gene for CCR5. This gene provides the protein which is an entry point for HIV into the cell. We know that one of the mutants for it confers resistance to HIV, and therefore deleting it would as well. The father was HIV positive, and the mother was HIV negative, and they had informed consent on the procedure. Take a moment and either individually or with your group, discuss this and discuss why it was so controversial. After all, it could certainly be argued that he was helping the girls by ensuring that they would not need to worry about being infected with HIV. One bit of background information that is useful here is that we do have protocols for washing sperm in a way that leads to virtually no risk of children getting HIV from their fathers. Pause now and come back when you've had some time to think about this or talk about it with your groups. So there are certainly layers of issues here. A more administrative one is simply that he didn't really have approval from basically anyone on this. Something this big is largely seen as something that deserves a bit of input from more than one researchers and the parents. That is definitely pretty far along the value judgment though, because who does have the right to regulate that? If not the parent and their doctors, does their country? But it's affecting the entire human population, so perhaps only a world agency? Yet we don't have a single world government. So this gets very tricky very quickly. So let's move on beyond the rogue scientist idea and talk about why we should give pause to this in general, still sticking largely to the more sciencey stuff. One of the biggest issues at play is that the CCR5 gene isn't purely an HIV transport protein. It does a lot of other things too. And based on its evolutionary history of being both very recent and incredibly common, it's actually pretty likely that there's some beneficial attributes that allowed it to be selected for in the population. We don't know what knocking this gene out really does, and that's pretty important to know before you start affecting people's lives with it. One of the other big issues that goes back up to this idea of about it being more than just a decision between consenting parents and their doctor is that he changed the germline. What this means is that the edits were made well before the eggs in the girl's ovaries had formed. Any babies they have will also have this mutation. It puts the edits permanently into the larger human gene pool, and this becomes a much bigger deal than a few biohackers injecting themselves with things that will not make it into the human gene pool. Which isn't to say that those biohackers won't have kids, just that they're not editing the cells that are actually responsible for donating the DNA to the babies. So take a moment and watch this video and then come back. Pause here, come back when you're ready. This clip of Star Trek starts to get at this idea, albeit in a bit more entertaining way than my last slide. Genes may largely code for just one protein. But that one protein may do a lot of things, and all of these things are super complex. So if you know that some genes play a role in intelligence and you alter those, you may also increase the chance for a particular mental illness, which is what they are kind of playing up here with making everyone who'd been engineered, except Bashir, seem a little insane. If you alter a gene that plays a role in reaction timing, you may accidentally change some other way that a patient responds to stimulus. Whenever we alter a gene that is serving one purpose, we need to account for every other purpose that gene may fulfill, both known and unknown to us, and it's very difficult to account for things that we don't even know. Please note that I am absolutely not saying that this means we shouldn't do it. I actually probably fall a bit more on the pro-CRISPR people side of the debate, honestly. I just think that we need to do it with full open eyes to what can happen, and to remember that the people that would be created from this didn't have any say in the matter. Those fetuses turned to babies turned to adults had no consent in their genetic editing. Take a moment and watch this video on gene drive. This takes a slightly different take on the germline idea, but some of the same issues come into play. And then come back when you're ready. Here we are using this in a very particular way to effectively kill off, or at least severely limit a species. This, of course, gets scary when you're manipulating an environment in a way that doesn't really allow for takebacks. But it could make a huge difference in saving humans and animals from mosquito-borne illnesses without having to constantly use pesticides, which could also kill off many beneficial species like bees. I don't think I have much to say about it other than that it's super cool. This gets talked about more in the Netflix documentary that I recommended before. They go pretty far into how the scientists work with communities to make sure that it's a good fit for them, and that they actually really want this to be done in their communities. It's a great example of how scientists need to be good communicators, and community leaders need to be able to work with and listen to scientists. Pretty much all the things that this class is about. All right, I know, a lot of videos to watch in this video. But this is a very cool, super fast evolving area, and everyone wants to talk about it. So I'd rather let you listen to a lot of the experts with a lot of the animations than just me talking on slides about it. So watch this, and then come back. Give some thoughts to these two questions here. Why can't we bring back the dinosaurs? And what extinction issues could CRISPR help out with? And then talk about them in your groups. This is going to be our last slide of this video. I'd like you to spend time talking through this with your groups before watching the end of the video. So think about which of these things may or may not be possible and why. If you want to start moving into ethics, go for it, but please don't only talk about the ethics. We want to get into some of the science stuff here too. So pause now and have fun talking in your groups, filling out your form, and then come back after you're all done with that. There's really nothing after the slides, so you're fine to pause this until after your group work. If your group does get stuck, you can also take time and watch this as a group and then discuss your thoughts on my thoughts on it as well, if you can't quite do it on your own. Whatever leads to the best conversations for you and your group members is great. So pause now, come back when you're ready. All right, let's just go through these in order. Giving pigs wings gets into a tricky concept of how many genes are required to make a thing happen. Pigs have never had wings in their evolutionary history, so knowing what genes to turn on, insert, or turn off would be incredibly difficult. Difficult to the point, perhaps, of impossibility. There are some in-between concepts here that might be more doable. For instance, we have had tails in our evolutionary history, so perhaps it would be possible to figure out where we differ from our tailed cousins, and maybe give humans tails. That's something that I'd put more on the incredibly difficult, but maybe possibly doable. Pigs with wings, maybe a bit tougher. Let's move on to the next one. The curing of basically any genetic disorder, where we know exactly what gene has gone wrong, I think actually is a very real possibility. The limitations here would be more related to how early can we do it, and has there already been damage done? We know that you can do it very early on in embryonic development, so we could definitely change it super early. That being said, if you didn't actually catch that there was a problem until halfway through pregnancy and there has already been developmental problems caused, that may have some limitations on it. Curing Alzheimer's is an interesting one. The easy thing here is to go after the genes that make you prone to early onset Alzheimer's. And making sure that people don't get this until they're well into their 90s or 100s is pretty close to a cure. However, you also need to deal with the environmental risk factors that gene therapy isn't going to fix. And then we don't know in some cases that are not related to the particular genes that we already know about or the environment, why some people still seem to get this earlier than others. So that would be a harder area of it to fix. We could fix the ones directly related to the genes that we know about, maybe not so much the others. Maybe it turns out those are actually related to a particular gene too that we just don't know about, or maybe 2 or 3 genes, and we could do something about it later on once we know that. But right now it would be a little tricky. And then of course, if we all start living into, let's say, our 110s, perhaps these tweaks will no longer suffice and we'll need something more, because we all do have those plaques that eventually turn into Alzheimer's. It's just they don't get bad enough to actually affect people in a large portion of the population. Reversing aging I tossed in here largely as a conversation piece. I don't really have great answers for you. We know that aging has a lot to do with environmental factors and genetics, but the genetics aren't clear. There is likely some typically thought of genetic markers that change aging as we know it. The very, very long-lived people tend to have family history of long lives. So when we're talking kind of our centenarians. But that isn't the whole story. There are probably some epigenetic factors which relate to what genes are turned on and off at different points in their life. So just to kind of repeat that, epigenetic is the idea that you can turn genes on and off, and we don't entirely know how that relates to aging, but we think it does. So that being said, CRISPR is probably going to be able to edit epigenetics as well. They're working on that too. So even if it is an epigenetic issue, we might still be able to use CRISPR for it. So if we do figure out what's going on with aging and how this is related, perhaps we could fix that. There is also a relationship to telomeres with aging, which are these kind of end caps to the DNA, if you want to think of them like that. And it seems that as telomeres get shorter, people get older. We don't entirely know that that's causative in that direction, though. There's still kind of some up in the air research there. That being said, if longer telomeres does mean that does reverse aging, CRISPR could probably edit that as well. That being said, if you want to know more about that, there's lots and lots of videos on telomeres, so you can go ahead and Google any one that you like. So at the core, our limitation here is not so much in our techniques of editing things, but in the fact that we don't really understand the aging process all that well. Even in the genes that we can track and we can say are changing during aging, those might be protective. It might actually be a response to getting old as opposed to causing getting old. And if we were to undo them, we could actually speed up aging or make it worse. So that's tough. We just don't have enough information. Once we do, though, CRISPR will likely hold a lot of answers for us. It provides us with tools to understand it in some cases, so it could also help us get there eventually. But it's probably a long ways off. I'm not really holding out a lot of hope that we'll be there in time to save me, but perhaps you guys, and if not you, maybe the generation below you. There's a link to a couple of videos in the notes if you would like to listen to more videos on this, one on epigenetics and one on telomeres, in case you want to learn more about that. If you're not interested in that, don't worry about it. Now, next one, making humans smarter. This has a similar issue to the last one. Though some genes do seem to be associated with cognitive function, we don't really know what makes people smart. Heck, we really don't even have a true definition or measure of what smart is. You can, of course, say, well, it's IQ, except that performance on IQ tests is really highly related to your socioeconomic status and the experiences, which makes it even trickier to sort out exactly what the genetic component is. This isn't to say that it isn't possible to change the heritable component of intelligence, but we don't have a good enough grasp on what that heritable components are to really know how to do this at the moment. So we might be able to make small changes here and there, but it probably wouldn't be anything too dramatic. Of course, as more research comes out, if it turns out there is a little bit more of a heritable component in other places that we hadn't found before, we might be able to edit that and change things. That being said, even if we change all of the heritable components of it, we still have to deal with the issues of not having the right nutrition or the right specific nutrients such as iron or zinc or other things that we're finding out really matter. And then of course the experiences of early on in childhood development that make a very big difference later on in life. So then lastly, we have these two surrounding endangered and extinct species. Dinosaurs are basically a no-go because we don't have their genes to copy. So the best that we could possibly do would be some sort of dinosaur-like creature made from our understanding of species that have survived since then, and then some evolutionary mapping. But it wouldn't be the same as bringing back the actual T-Rex. It could play an important role in newly extinct or endangered species, though. We might be able to edit the genome of a species to help it survive something that is killing it off, perhaps giving it a benefit to a changing climate or diet. It could also be a way to increase genetic diversity in severely bottlenecked species if we have samples from others that would help us to understand how to safely edit their genome. With any of these, we can, of course, start to delve into the should-we ethics of all this. Some of it seems pretty straightforward. Curing diseases in patients, for instance. Though what is a disease? What happens when this definition is broadened? If we get a better understanding of genetic component of autism, should we start to eliminate that? What would that do to something like the field of science, which has a very high population of people with autistic traits, even if they don't have an actual diagnosis? People often talk about designer babies as if choosing hair color is a problem. But what if you can change their intelligence? Should we allow for that? What does all of this do to the diversity of human traits? After all, if you make them smarter, wouldn't it also make sense to make them more emotionally stable, less prone to depression? All of these traits make up who we are as people, for better or worse. If we could reverse aging, should we? What happens to our world and our economy if suddenly 120 is the new 80? If suddenly instead of having 20 to 30 years of fertility, we have 50 or 60? What does that do to the world population? And then who has access to that technology? Even if we just stick to animals, it can get messy. Should we use our technology to try to fix the mass extinction events that we have created? Are we sure enough of our abilities to help one species without harming a whole series of others? This is, of course, just a sampling of possible applications and limitations of CRISPR. Next class, you'll learn how our history of eugenics can inform some of our thinking on CRISPR.